to the PAD podcast. I'm Shafir Rahman, your host, with new exciting episode. So stay tuned in. Please follow me on YouTube, Twitter and Instagram with the handlebar at Shafir Rahman 4109. And please feel free to share these chunks of knowledge. Thank you. Today, the topic of our discussion of the podcast is Parkinson disease. Now, before discussing the Parkinson disease, first of all, we have to understand the word Parkinsonism. So, Parkinsonism is an umbrella term under which Parkinson disease comes, and it also includes several other diseases which we will discuss shortly. So, Parkinsonism is a clinical syndrome characterized by tremors, rigidity, bradykinesia, and instability. Now, these types of motor disturbances may be seen in a range of diseases that damage dopaminergic neurons, which project from the substantia nigra to the striatum. Now, Parkinsonism can be induced by drugs such as dopamine antagonists or toxins that selectively injure dopaminergic neurons. Now, among the neurodegenerative diseases, most cases of Parkinsonism are caused by Parkinson disease, which is associated with characteristic neuronal inclusions containing alpha-synuclein. Other diseases in which Parkinsonism may be present include multiple system atrophy, in which alpha-synuclein aggregates are found in oligodendrocytes. Then there are two other diseases like progressive supranuclear palsy and corticobasal degeneration, which are both associated with tau containing inclusions in neurons as well as in glial cells. And then the last one is the post-encephalitic Parkinsonism, which was associated with 1918 influenza pandemic. So it means that Parkinsonism is an umbrella term, and under that umbrella term, you will find 5 different diseases. And one of that is the Parkinson disease, which we will discuss today. But it's not only the Parkinson disease which is included under the umbrella of the Parkinsonism. We have multiple system atrophy, progressive supranuclear palsy, corticobasal degenerations, and post-encephalitic Parkinsonism, which are included under Parkinsonism. So, Parkinson disease includes neurons having inclusions containing alpha-synuclein. Then multiple system atrophy contains alpha-synuclein aggregates in oligodendrocytes. Then progressive supranuclear palsy and corticobasal degenerations contain tau containing inclusions in neurons and glial cells. And post-encephalitic Parkinsonism was associated with 1918 influenza pandemic. Now let us try to discuss the pathogenesis of Parkinson disease. Now, most cases of Parkinson disease are sporadic, but autosomal dominant and recessive forms of the disease also exist. So, point mutations and duplications of the gene encoding alpha-synuclein, which is a protein involved in synaptic transmission, cause autosomal dominant Parkinson disease. Now, even in the sporadic Parkinson disease, the diagnostic feature of which is the presence of the Lewy bodies, which is an inclusion containing alpha-synuclein. So, the linkage between alpha-synuclein and disease pathogenesis is unclear, but other genetic forms of Parkinson disease provide some clues. Now, 2 other causative genetic loci encode the proteins Parkin and E3 ubiquitin ligase and UCHLI, which is an enzyme involved in recycling of ubiquitin from proteins targeted to the proteasomes, which suggests that the defects in protein degradation may have a pathogenic role in the progression of the Parkinson disease. Another clue comes from the association of Parkinson disease with mutations in protein kinase called LRRK2, and when there is a mutation of LRRK2, so when we see the tissue under histopathological examination, so it will show Levy bodies containing alpha-synucleins or tangles containing tau. And finally, some forms of the familial Parkinson disease are associated with mutations in PARK7 and pink eye genes, both of which appear to be important for normal mitochondrial functions. Now, just we will briefly recap the pathogenesis so we should understand much more about it clearly. So look, what I've said that both sporadic and familial form of Parkinson disease exist. Now, alpha-synuclein, which we were discussing, is a protein that is involved in the synaptic transmissions. So point mutations and duplication of the gene encoding the alpha-synuclein cause autosomal dominant Parkinson disease. Then there is another entity which is important in Parkinson disease understanding is the presence of the Levy bodies, which we will discuss in much more detail in the morphology section. But Levy body generally is an eosinophilic. Eosinophilic means that histological sections that take up the staining dye eosine and which imparts red or pink color. So it's basically an eosinophilic cytoplasmic inclusion consisting of dense core surrounded by a halo of 10 nanometer wide radiating fibrils, and the primary structure of which is alpha-synuclein. So the fibrils, the primary structure which consists of that fibrils is alpha-synuclein. So sporadic Parkinson disease usually is associated with Levy body, and Levy body is a diagnostic feature of the sporadic Parkinson disease. The linkage between alpha-synuclein and disease pathogenesis of Parkinsonism is unclear. Then we have to know some genetic loci that encode like protein parkin, E3 ubiquitin ligase, UCHLI enzymes. So when there is some sort of mutations or some sort of genetic defects in these proteins, so Parkinsonism can emerge. So based upon this theory, this theory suggests that defects in protein degradation pathways may play an important role in the pathogenesis of Parkinson disease. Then there is another association of protein kinase LRRK2, and LRRK2, when there will be mutations in the protein kinase LRRK2, so it may be associated with Parkinson disease. And on histopathology, when there is LRRK mutation, you will see Levy bodies containing alpha-synuclein or tangles containing tau. Then the familial Parkinson disease is also present, in which there are mutations in PARK7 and pink eye genes, and these genes are important for normal mitochondrial functioning. Now let us discuss about the morphology of the Parkinson disease. So a typical gross finding at autopsy is pallor of the substantia nigra and locus ceruleus. So substantia nigra is a structure present in the basal ganglia in the midbrain, and locus ceruleus is the nucleus in the pons of brainstem. So first thing is the gross finding is the pallor of substantia nigra and locus ceruleus, and microscopic features will be the loss of the pigmented catecholaminergic neurons plus gliosis. So when there will be the loss of the pigmented neurons, which are dopaminergic and catecholaminergic neurons, so what will happen that the color will be the pallor, there will be yellowish tinge to the structure because there will be the loss of the pigmented neurons. Included with that will be the gliosis, which is a non-specific reactive change of glial cells, for example microglia, in response to the damage to the CNS. So whenever there is a damage to CNS, so there is a non-specific reaction which is known as gliosis. So gliosis will be also be present in the morphology of the Parkinson disease. Then, as we have discussed about the Levy bodies, we have to understand it much more clearly that it's basically a single or multiple intracytoplasmic eosinophilic round to elongated inclusions that often have a dense core surrounded by a pale halo. And what it is consisting of? Like on ultrastructure examination, Lewy body consists of fine filaments densely packed in the core but loose at the rim, composed of alpha synuclein, neurofilaments, and ubiquitin. Then there is another structure which is present in the morphology is the Levy neurites. So neurites or neuronal process refers to any projection from a cell body of a neuron. So Levy neurites are dystrophic neurites that also contain abnormal aggregates of alpha synuclein. So as implied by the occurrence of a broad array of neurological deficits in Parkinson disease, immunohistochemical staining for alpha-synuclein highlights more subtle Lewy bodies and Lewy neurites in many brain regions outside of the substantia nigra and in non-dopaminergic neurons. These lesions appear first in the medulla and then in the pons before involvement of the substantia nigra. So, as implied by the dementia, Lewy bodies and Lewy neurites eventually appear in the cerebral cortex and subcortical areas, including the cholinergic cells of the basal nucleus of the mynarid and the amygdala. So, again, recapping the morphology of the Parkinson disease, that first of all, we have to know the gross findings. The gross findings, you will see the pallor of the substantia nigra and locus ceruleus. On microscopic examination, you will see the loss of pigmented catecholinergic neurons, and also superimposed on that will be the gliosis. Then you have to understand the structure of the Lewy bodies, that what Lewy bodies are, which are basically intracytoplasmic eosinophilic round 2 elongated inclusions, and it is composed of like alpha synuclein and neurofilaments and ubiquitin. And then you have to understand the concept of the Lewy neurites, which are dystrophic neurites and also contains abnormal aggregates of alpha synuclein. So now we will discuss the clinical features of the Parkinson disease. So Parkinson disease commonly manifests as a movement disorder in the absence of a toxic exposure or other non-underlying etiology. The disease usually progresses over 10 to 15 years, eventually producing severe motor slowing to the point of near immobility. And that usually is the result of intercurrent infection or trauma for frequent fall caused by the postural instability. So movement symptoms of the Parkinson disease initially responds to L-DOPA, also known as L-dihydroxyphenylalanine, but this treatment does not slow the disease progression. And over time, L-DOPA becomes less effective and begins to cause potentially problematic fluctuations in motor functions. Now, while the movement disorder associated with the loss of nigrostriatal dopaminergic pathway is an important feature of Parkinson disease, it is clear that the disease has more extensive clinical and pathological manifestations. Lesions can be found lower in the brainstem in advance of nigral involvement, in line with the clinical studies showing that autonomic dysfunction and behavioral disorders often are present in the advance of the motor problems. So dementia, typically with a mildly fluctuating course and hallucinations, emerges in many persons with Parkinson disease and is attributable to involvement of the cerebral cortex. So when dementia arises within 1 year of the onset of motor symptom, it is referred to Lewy body dementia. So word about Lewy body dementia versus Parkinson disease. So we have to understand both of them. Like in Parkinson disease, you will also find Lewy bodies, and in Lewy body dementia, you will also find Lewy bodies. So both related to the abnormal deposits of protein alpha synuclein. Both have similar symptoms of cognitive decline, movement problems, and hallucination. But the main difference between both of these diseases is the order of the onset of the symptoms. Now, in Lewy body dementia, cognitive symptoms develop within a year of movement symptoms, or sometimes even before. So that is 1 year cutoff point, that if after movement disorder within 1 year there are cognitive symptoms, for example dementia, so what will happen that when it is within 1 year time span or earlier than that, we will say that it's a Lewy body dementia. But if it is later than 1 year or much Later than that, so you will say that it's a Parkinson disease. So in Parkinson disease, cognitive symptoms develop more than a year after the onset of movement symptoms. So this concludes our podcast for today. For further questioning related to the topic, feel free to email me at shafirehman at hotmail.com.